Okay. So the quiz on Wednesday will be over the chapters 20 and chapter 21 on the
last week's and then today's.
Yeah, and today's. Okay, so the last thing that we were talking about in terms of the immune system were vaccinations. And then, so you learned that you can be presented with a pathogen that has been weakened or killed, or maybe even just a piece of a pathogen, or in the case of things like tetanus, the toxin that a bacteria produces, right? And then you are able to learn how to fight off that infection. So that is what we refer to as active immunity. So active immunity is a person's own immune system is producing the antibodies. Okay, so that would include vaccination, right? Because you're still activating a person's immune system so that they'll produce antibodies. They're learning, they get memory cells. But it also includes exposure. So people that are exposed to, say, for example, chickenpox virus generally do not get it twice, right? Their immune system learns how to fight off that infection. Now, with the chickenpox virus, it's kind of bad because that virus tends to stay latent, dormant in the body. And what does it cause later on if your immune system is compromised? Shingles, right? So it can cause shingles. If you become stressed out and your immune system's depressed, or if you get AIDS and your immune system's suppressed, or if you get older, then you can get shingles, okay? So that would be active. So passive would include individuals obtaining antibodies from another source. Okay. So what is an example of passive immunity, do you think? Where you obtain antibodies from another source. How about breastfeeding? Okay, so breast milk. And there's a name for the first type of breast milk that is produced, and does anybody know what that's called? Colostrum. Okay, so colostrum, I remember how to spell it. Oops, that's not how you spell it. That's how you spell it? I think that's how you spell it, colostrum. Anyway, colostrum looks very different from milk that is produced later on. So it's kind of watery and it's yellow and it is just full of protein. So it's full of antibodies. So the idea here is that a mother is going to be exposed to all of the different pathogens in the environment that the potential baby is going to be exposed to. Thank you. One of those days. Okay. That's why breast milk is so important to the immune system of the baby early on, right? So colostrum. The breast milk also contains antibodies later on where it becomes more fat-rich and full of nutrients for baby's growth, but it just looks a little bit different. So it's more mature, but still has antibodies. Another example of this would be when you take antivenoms. Right? So if you get bit by a rattlesnake, for example, that releases a neurotoxic into your body. And so you can go to a hospital and they will have antivenoms. Some hospitals even have antivenoms for spider bites, right? And that will help to, or that does contain antibodies that will bind to the toxin and inactivate it so that it doesn't cause death, okay? Another example is what is referred to as gamma globulin shots. So gamma globulins are antibodies, a specific class of antibodies. They're called IgG antibodies. We're going to talk about that in a second. So those are immunoglobulin G. Has anybody ever had a gamma globulin shot? Okay. So when I was a kid, I was sick all the time, and this is really weird, but my doctor decided that I needed gamma globulin shots to keep me from getting sick all the time. And so like every 2 weeks I'd have to go, and they would essentially inject antibodies in me to try to keep me healthy. So they don't really do that anymore, probably thankfully, because it was actually a really painful injection, and it was really awful. But anyway, but they do this sometimes in the military. So imagine if you have a bunch of troops that are going into an area where they have not been exposed to the viruses and the bacteria and all the other things that can potentially make them really sick when they get there. And so you wouldn't want them to like fall down and start vomiting and having diarrhea, the whole troops, right? So sometimes you get, they give gamma globulin shots to soldiers that are going into a particular area so that they have the antibodies that will help them fight it off, okay? The thing about this is that all of this is short term, right? Whereas the active immunity is long term. So with long term, you get the production of memory cells that are specific to the specific pathogen. And these antibodies eventually just break down, right? And you would not produce your own antibodies after those antibodies that have been injected into you are gone, okay? So that's the difference between passive and active. And everybody always gets this wrong, so you might want to star this, that vaccinations are active. So for some reason, whenever I ask this question on a quiz or a midterm or a final, half the class will put, oh, it's got to be passive because you were given it, right? But it is still active because your own immune system is learning how to produce the antibodies, so it's still active immunity, okay? Okay, so let's look at these antibodies in more detail. And there is a mnemonic device to remember the types. So there are 5 classes of antibodies. And this is abbreviated as MAG. Oops, MAG. So there is IgM, IgA, IgD, IgG, which I just mentioned, and IgE. So the Ig stands for immunoglobulins. So that's Ig. So just think about globular proteins, right? And they have an immune function. So they're called immunoglobulins. This is the exact same thing as antibodies. So you can use antibodies and immunoglobulins interchangeably, okay? So if we look at the types, we can have this should be Ig, sorry. IgM, IgA, IgD, right? IgG, and IgE, okay? So IgM is a really interesting antibody because it's the only one that is large because it's made of 5 Y-shaped molecules. So it's actually a pentamer, which means that it's 5 Y-shaped molecules hooked together. And this type is found in the blood plasma after you are initially infected. So it's important for the primary response to infection. So it's your body starts pumping out after it senses an invader, it starts pumping out IgM. And it is also important in agglutination of red blood cells. So remember that we looked at during our blood lab, right? We looked at how when you have antibodies present, it will cause the red blood cells to clump. and then you could test your blood type, but it would also be very bad if the agglutination, which is different than a coagulation, occurred during a blood transfusion. Okay, so if we look at a picture of this in your book, the IgM, okay, Y-shaped, and it looks like this. Okay, so here, there would be like a red blood cell here attached, right? It attaches to the red blood cells and it causes them to clump. Okay, so that's the pentamer. Now, the other one, well, the next one, MAJ, is A, and it's a dimer. So it has two Y-shaped molecules bound together. And this one's not really found abundantly in the blood, but it's found in breast milk. So it's the predominant one in breast milk. It's also found in the saliva and tears and sweat. Okay, so body secretions. So IgA is in body secretions. So it would be like saliva, milk, which is a body secretion, tears, sweat, and intestinal juice. Okay. So that's IgA. So the next one is IgD. And IgD, all the rest of them are just single Y-shaped molecules. So these are all what are referred to as monomers because they have just a single Y-shaped molecule. So the IgD is important because this is the type of antibody that is found as a receptor on B cells. So this would be as a receptors. These are receptors on B cells. So remember that B cells are a type of lymphocyte that have their own antibody receptors. We're going to talk about T cells next, which are the other type of lymphocyte that lack receptors. And you saw that in the video that we watched in lab. Okay. Now, gamma globulin shots, IgG. This is the most abundant antibody in your blood, right? And so this is really important in the late primary response. And it also crosses the placenta. And so it provides and confers some immunity to the fetus. So even as the mother is has the fetus inside of her, antibodies can cross, right? Now, what was the problem with a female's blood type? What blood type did you have to worry about that she could possibly produce an antibody that would attack the fetus as it was developing? Do you remember from our lab?
The mom is negative and the baby's positive.
Right, so Rh negative, right? So if you are a female and you have type negative blood, oftentimes they will give you an injection of a drug called RhoGAM. And what RhoGAM does is it binds those antibodies and, it binds the antigens and it actually prevents the mother from producing the antibodies and learning to fight off those, okay? So IgG is most abundant in the plasma. Right? And it crosses the placenta. Right? And it is the type of antibody that they give in the gamma globulin shots. Okay. So the last one, IgE, you can kind of remember E's as being like eosinophils. Remember those cells? But E means, for some reason, they've given the E is the one that is associated with parasites. And so you can kind of remember eosinophils are white blood cells that attack parasites, and IgE are specifically the type of antibody that's produced to fight off parasitic infections, and also these are the ones that cause allergic responses, okay? So parasitic infections, okay, and they cause allergic responses. And we're going to talk more about the allergic responses in a little bit. So generally, you do not have a lot of IgE unless you're having a severe allergic attack, or if you have chronic parasitic infections. So if you have lots of IgE antibodies, then that is what you would have in your body. Okay, so let's look at the mechanisms, the way that the antibodies act. And you saw some of that in the video that we watched in lab. And I like to use the mnemonic device PLAN here as my remembering how antibodies act. Okay, so MAGs have plans, right? Okay, so P stands for precipitation. So let's say you have a toxin in your blood and it is in solution and it is traveling throughout your bodies and getting into your nervous system and causing horrible things happening. So if you take that toxin out of solution, then you precipitate it, right? So this takes antigens out of solution. So they cannot be transported effectively in the blood or in the fluids surrounding the tissues. So that's what precipitation means. Okay. Next, we have lysis. And we talked about a group of proteins that were actually part of the innate immune system that help in lysis. And do you remember, they weren't the interferons. What type of proteins that do you have in your blood that help in lysis? Complement proteins, okay? So this enhances complement protein function. And there's actually a name for this enhancing, and it's called opsonization. Okay. So when the antibodies attack and attach to a particular pathogen, for example, that attracts complement proteins, and that attraction of the complement proteins is called opsonization, right? And then what happens is that the complement proteins insert themselves into the membrane of the bacteria, for example, and then the bacteria, water will flood in and the bacteria will burst, or lysis, okay? So in your book, they have this picture. So this would be precipitation, taking them out of solution, right? This would be activating the complement system, right? So the binding of the antigens helps in that lysis, right? And then we also have A, which is agglutination. Okay, so A stands for agglutination. And here you can see the pentamer IgM antibody or immunoglobulin adhering to different red blood cells, right? And so that would be an example of agglutination. Okay. And then we have neutralization. So that would be making the pathogen or the toxin or whatever it is that's foreign in your body not harmful. So it decreases the harm of the bacteria. So the bacteria maybe can't reproduce. Maybe they're unable to produce toxins that destroy tissues, right? So it neutralizes them. And so here they're showing would be neutralizing a part of a virus that would prevent the virus from infecting its host cell. And then all of these things will enhance phagocytosis. So this was a type of macrophage that is ingesting the antigen and cleaning it up, right? And then also inflammation, which would be the swelling of blood vessels and allowing the neutrophils to move out into the tissues. Okay, so that's the idea that the antibodies have a series of things that they can do to enhance the immune system. Okay, so we talked about humoral-mediated immunity or antibody-mediated immunity. Now we're going to talk about cell-mediated immunity, and these are what the T cells do. So these are the T lymphocytes in your body. So these are lymphocytes. Right? They lack antibody receptors. They must be presented with antigens. And the cells that present the antigens are called antigen-presenting cells, or APCs. These are antigen-presenting cells. So the APCs could be white blood cells. So they could be, for example, neutrophils. We could also have macrophages as presenting cells. We could also have cells that are called dendritic cells. And so in the skin, the dendritic cells, and in the membranes, like in the intestine, the skin, those membranes that are in the lungs that are close to the outside, it's the dendritic cells. And dendrite it means branching. And so these cells have lots of branches. And so these are some examples of antigen-presenting cells. Okay? But the other cells that can present are just body cells that have been infected or that have become cancerous. So infected with a virus or a parasite, right? That says infected, let me spell it better. Infected or cancerous. Okay, so the thing about presenting to a T cell is that you have to use, we already talked about this, you have to use proteins that are on the surface of the cell, and they're called MHC proteins. So they're major histocompatibility proteins. So these neutrophils and macrophages and dendritic cells, they use MHC proteins, and specifically class 2. Okay. So there's two different types of classes. There's class 1 and class 2. So they have a special MHC protein on them, and it's class 2. And they present exogenous antigens. So exogenous means that they have come from the outside, right? So it would be like a macrophage eating a bacteria, taking a piece of that bacteria, which would be an exogenous antigen, and then using its class 2 MHC protein to present it to a helper T cell, okay? So if we look at these cells, all of our cells have on them MHC proteins. And so this is the MHC protein class 1. Right? So all of our cells have the ability to present. And they might not be doing it purposefully. It could be that they have become cancerous and their DNA has changed and the proteins that they're producing change. And so they just kind of like are producing weird proteins that are ending up on the surface of them. And then our white blood cells can see that these are strange proteins and then attack the cancerous cells before they cause damage, okay? So these present what is referred to as endogenous antigens. So endo means inside, exo means outside. So this would be abnormal proteins, abnormal self proteins. Okay, so they're producing proteins, these cells are producing weird proteins because they're either infected or they have become cancerous. Okay, so that's MHC2, or class 1, sorry, class 1. So in your book, they have these really nice diagrams that show the difference between the class 1 and class 2. Okay, oops, okay. So this is my endogenous protein, right? So this could be produced by, let's say it's a cancer cell. So the cancer cell produces this strange protein, and then it gets presented on its surface using the MHC class 1, right? So that would be an example of an endogenous antigen being presented. And then this is an example of a white blood cell that has taken in something from outside, so it becomes exogenous antigens, breaking it down, and then presenting it on the surface. So that's the MHC class 2, okay? So that would be an example of an exogenous piece of protein that is being presented to a cell, okay? Okay, so let's look at T cells. So remember that T cells are produced randomly, right? And they have to be filtered, the ones that could possibly attack your cells or that could not recognize self-antigens, so that we have selection of them. So this is very much like the clonal selection of B cells, right? So we have randomly produced T cells that are able to recognize different antigens. Okay. So let's say one of these cells has been presented with an antigen and it recognizes it. Okay. So we have a type of T cell that's really important in this, and they're called helper T cells. Sometimes this is abbreviated as a big T with a little h underneath. So these were the lymphocytes that you actually got when we were playing the immunoscenarios game, when you got the little blue card that showed the helper T cell that was specific to a particular antigen, okay? So the helper T cells do a couple of things. They undergo mitosis. Remember, mitosis is the type of cell division that clones, right? So they produce clones of themselves, and they produce memory T cells. Okay, so the helper T cells undergo mitosis, and so this is the clones, right? They also release chemicals. So they release cytokines. So cytokines are chemicals that signal B cells and another type of cell called a cytotoxic T cell. Okay, so cytotoxic T cells are sometimes rewritten like this, with a little T with a little C underneath. Okay. So these cytokines are really important for coordinating the humoral immunity and the cell-mediated immunity. Humoral immunity is B cells. Cell-mediated immunity is the cytotoxic T cells, right? So this is cell mediated immunity. Okay. Meaning that these T cells, unlike the B cells, will actually release chemicals that will kill microorganisms and also cells that have become cancerous or infected with the virus. Okay, so if we look at this process, it looks very similar to the handout that I gave you last Wednesday. Okay, so this would be an example of a lymphocyte. Okay, here I am activating my helper T cells. So notice that my helper T cell that is specific for that MHC protein and is being presented with it will undergo cell division. It'll produce memory cells, right? And then it produces these helper T cells. And the thing that I wanted to point out is these helper T cells are also really important in, let's see, activating immunity. Can I see that picture? I have a picture that'll show that in a second. Okay. Okay. So we can also have the same type of clonal selection here with the activation of the cytotoxic T cells. Okay, so clonal selection just means that the T cell recognizes its antigen and then undergoes cell division. It's selected and undergoes cell division. Okay, so we already talked about helper T cells, sorry. Okay, so let's talk about AIDS. Now, AIDS is short for acquired immunodeficiency syndrome. Underline acquired a couple of times because for some reason you think A and you think autoimmune deficiency syndrome, and that's a totally different thing. So acquired means that it, and is there a C in acquired? ACQ. Is C? I'm not sure. Okay, it's like acquired. So, okay, acquired immunodeficiency. Okay, so it is acquired from a virus. And so it is caused by the HIV, which is a human immunodeficiency virus. Okay. So we know that this virus specifically attacks helper T cells. And it uses them to reproduce. So it's unlike other viruses that might attack your respiratory system. It might attack your liver, like hepatitis viruses attack your liver. This one specifically attacks helper T cells. And it attacks them by using the receptors on their surface to gain entry. So it uses CD4 receptors to gain entry. And that'll be important because we're going to talk about some individuals are actually have a mutation that changes this protein so that the virus can actually not get inside of it. Okay. Okay. So one of the really interesting things about AIDS is that we say that it has a long latency, right? So that means like how long can you have the virus before actually getting AIDS? So this could be like 5 to 20 years, maybe even longer, right? So we don't know. There might be people that could have the virus for like 30 years, since it was, actually it could be coming up, because it was 1980s that we discovered it, right? That would still have the virus but would not actually get AIDS, Okay, so that has a long latency. It also has a low or a weak, it's relatively weak, and so it has a low virulence. So what this means, virulence, is how sick it makes you over time, right? So because it doesn't make you sick, really sick initially, it has a low virulence compared to other viruses like H1N1 or like Ebola, right? Ebola would have a short latency and it would be highly virulent, right? And so I'm going to give you a handout, and because I didn't have my key card, I wasn't able to, I'm going to have your guys' quizzes too. I wasn't able to punch holes in it. This is the handout. So this is showing what is happening over time to a person that has been infected with HIV. And so if we look at it, okay, so within 1 to 2 months, you'll notice that the HIV count peaks in the blood, okay? And then what happens is the helper T's come in, and they're actually able to very dramatically decrease the count, right? Because they haven't it hasn't been that long and they haven't been affected that much, right? And so what do you think that this count is, this rapid decrease in HIV is due to? What are the cells producing, you think, that helps fight off HIV? Humoral immunity, so they're producing what? B cells, and then what? Antibodies, right? So notice that like your antibodies would be being produced, right? And they would help to fight off the initial infection. So it's really important to realize that the way that we test for HIV, we're not looking for the virus itself, right? We're actually looking for the antibodies. So testing of HIV this looks for the presence of specific antibodies. So for antibodies specific to the virus. Right? So it's not actually, viruses are so small, we don't actually see the viruses, right? But we can test to see if the antibodies are present, right?
Would the interferons be working with this as well?
Yes. So your interferons would also be working, and then also your cytotoxic T cells would be coming in and trying to destroy. So you have all the other complement parts of the immune system working, okay? So then you notice that at 5 years, or maybe 4 years, something interesting happens. So at 4 years, your white blood cell, the level of your T cells decreases below the HIV count, right? And so now, and there's different scales here, but now once a person has their helper T cells decline to a significant level, then their HIV increases, and then their immune system becomes severely compromised, right? And so this would be when a person starts to get sick, really sick, from like even things like the common cold, right? They get pneumonia, they get tuberculosis, they also get things like cancer because their immune system is not able to fight it off. So death, generally we say that death is not directly caused from AIDS, but it's caused from diseases that the body cannot fight off, right? So death results from disease that the body is not able to fight off. right? So that would be things like pneumonia, right? Cancer, big one. And then things like tuberculosis, which is at a greater, tuberculosis we fought off, and tuberculosis is caused by a bacteria in the lungs. But people that have HIV are more likely to harbor tuberculosis, and so they're more likely to be susceptible to it if it's present in the environment. Okay, so if we look at how or why HIV has such a low virulence and a long latency, we can talk about the evolution of virulence. So we'll talk about the evolution of virulence. And so when we talk about the evolution of pathogens, we call this coevolution. So this is how the host and the pathogen change in response to one another. And so I'm going to show you a short video that talks about this, and it talks about two examples. One is a bacteria, which is called cholera. And the cholera produces a toxin that causes diarrhea. And it's kind of in the news because of the cholera epidemic that's believed to be imminent in Haiti because of the recent hurricane. Right? So cholera, and then the other one is FIV, which is feline immunodeficiency virus. So we can look at how cats over time have responded to a virus, FIV, which we actually vaccinate our household cats against now. We can vaccinate them. But how the wild cats responded to it, okay? So I want you to watch this video and think about under what circumstances are pathogens becoming more dangerous, more virulent, with a higher, faster response, and under what circumstances they become less virulent.
If we can drive microbes to evolve drug resistance, then we can also make them evolve in ways that benefit us. This is the radical proposition of Amherst biologist Paul Ewald.
When people are looking at the antibiotic resistance problem, they see evolution as sort of the bad guy. It's the evolutionary process that's led to antibiotic resistance, and that's true. But just as easily, we can have evolution being the solution. In other words, we can have evolutionary processes leading to organisms becoming more mild.
Disease organisms evolve to be more or less harmful depending on how they are spread. Microbes that depend on close contact between people tend to be mild.
The rhinovirus that causes a common cold is transmitted by people walking around sneezing or coughing on other people. Since it really does depend on fairly healthy people to be transmitted, not surprisingly, it's one of the mildest viruses that we know about.
But microbes that are transmitted by insects or by tainted food or water tend to make people very sick.
The worst of all of the diarrheal bacteria that we know of have been waterborne. The bacteria that cause cholera and typhoid fever are often waterborne. So even if the organism is so harmful that the sick person can't move from bed, the organism can still be transmitted to large numbers of people. once we understand the factors that favor increased harmfulness and decreased harmfulness, then we can look at all of the things we do in society. We can ask the question, are we doing certain things, or can we do certain things that would favor organisms evolving towards mildness? We can look at the cholera outbreak in South America as a kind of natural experiment that allows us to evaluate these ideas.
In 1991, cholera invaded Peru and spread quickly. Over the next 5 years, more than 1 million people were stricken with diarrhea and vomiting, some severely. Over 10,000 people died. The disease was transmitted through water contaminated with human waste, or through food that was washed or handled by infected people.
There's a significant
Ewald collected strains of cholera bacteria from South America and measured the amount of toxin they produced, an indication of their virulence. Over time, he would document evolution in action.
If you have contaminated water allowing transmission, we expect the cholera organism to evolve to a particularly high level of harmfulness, and that's exactly what we see. We find that bacteria that had invaded countries with poor water supplies evolved increased harmfulness over time. They've actually become more toxigenic. They produce more toxin than they did at the outset. If instead we clean up the water supplies, then we force the bacteria to be transmitted only by routes that require healthy people. And what we find is that when cholera invaded countries with clean water supplies, the organism dropped in its harmfulness. Those bacteria evolved lower levels of toxin production. They actually became more mild through time. People would still be getting infected, but the infections would be so mild that most people won't even be sick. So the cholera outbreak in Latin America suggests that we may need only a few years to change the cholera organism from one that would often kill people to one that hardly ever causes the disease. What we're suggesting here is that we can domesticate these disease organisms very much in the same way that we've domesticated other organisms that are potentially harmful. For example, wolves have been harmful to us throughout our evolutionary history, but through domestication, some wolves have evolved into dogs that instead of harming us, actually help us. And I think we can do the same thing with these disease organisms.
Working with evolution instead of against it, we might eventually subdue even the deadliest microbes. Evolution has already forged such surprising truces.
Okay.
Most wild cats have evolved a way to live with a virus closely related to one that is decimating humans. The story was unraveled by Stephen O'Brien, here at the National Zoo to examine a tranquilized cheetah.
Well, we originally became interested in the cats because I was interested in the interplay between infectious diseases and the genes of the species that suffer them. We began working with cheetahs and subsequently started to study each of the 37 different species in the cat family. What we're learning from them is that they are mirrors of evolutionary processes in humans.
It all began in the 1980s, when O'Brien became concerned that small populations of endangered cats were especially vulnerable to the ravages of infectious disease. Then he heard that domestic cats were falling prey to a newly discovered and lethal virus, the feline immunodeficiency virus, or FIV.
FIV is associated with very skinny and malnourished and wasting disease in house cats. And that disease was the result of the collapse of the immune system. So the parallels with human immunodeficiency virus were very strong. I was curious as to whether or not the virus had also been able to infect non-domestic cats.
O'Brien had collected biological specimens from thousands of wildcats around the world. He began to screen them for the presence of the virus.
Well, when we did that in cheetahs from East Africa and the pumas in the Rockies and the ocelots down in the Andes and the lions in the Serengeti, it turned out that virtually every species of cats on the planet had been exposed to and infected with a version of feline immunodeficiency virus. Well, I was terrified because I thought that we were just a heartbeat away from an epidemic that was going to decimate some of these cats. And since 36 of 37 of these cat species are already considered endangered or threatened, then this could be the final wallop.
For years, O'Brien feared the worst. He urged zookeepers and game wardens around the world to test their animals for the virus and to watch for AIDS-like symptoms.
What we discovered, though, over time, is that these cats were really not getting ill. It was as if they had somehow come up with a resistance to a fatal virus.
O'Brien's research suggests FIV first infected the cat's ancestors around 1 million years ago. It decimated the animals. But a few cats carried mutations that made them resistant to the virus. These survivors passed on their protective genes to their offspring and to most wildcats alive today. Over time, the virus may also have evolved into less lethal strains. Today, wildcats and FIV have reached the end of a long evolutionary process and have adapted to each other. Humans and HIV only recently embarked on the path that might eventually lead to a truce. But the example of the wild cats convinced O'Brien there must be people endowed with mutations that protect them from HIV. He set out to find them.
Over a 10-year period of time, I quietly collected blood samples from 10,000 individuals that are high risk. My colleagues and I extracted the DNA, and we were stunned to discover a whopping mutation which protected against HIV infection. And it was the first gene that we could definitively say was influencing the outcome of exposure to this deadly virus.
Most people have receptors on their immune cells that allow HIV to dock and gain entry. But people with the mutation discovered by O'Brien lack some or all of these receptors. Infection by HIV becomes impossible. The mutation is present in about 10% of European Caucasians, but completely absent in Native African and East Asian peoples. Something in the evolutionary history of Caucasians must have favored the survival of people with this mutation.
We've actually used precise dating techniques to date the last time such a selective pressure took place, and that came out 700 years ago. Well, if you look in history books, that was the time of a rather dramatic infectious disease pandemic, which was the Black Death or the bubonic plague. And at that time, 1/3 of Europeans were wiped out.
A mutation that saved people from the plague 7 centuries ago may now protect their descendants from infection by HIV.
Today, when we scroll through the genes of cats or humans, we discover that they're littered with these footprints of historic epidemics that have defined the survival of today's living species.
We all bear the marks of our ancestors' struggle for survival. But evolution.
Stop there. Okay. So in terms of the cholera, what factors determine whether the cholera is going to become more virulent or less virulent? Dirty water. So maybe he referenced the ease of transmission, right? So if there's no way to get clean water, then you would expect that people are going to be drinking it, and then they're going to be getting the disease, and it's going to pass back into the water system because of lack of sanitation, and then it's going to become more virulent. So that's why they're so worried about outbreaks like in Haiti, because there's no time when we can say that the water is going to be clean enough, right? It's going to be really hard to set up a source of clean water that's not tainted with bacteria. So they would predict that the cholera over a short period of time will actually become more toxic under those circumstances, okay? So if you look at the HIV virus, it is not easily transmitted, right? So you cannot easily get it from somebody unless you have close contact with them. So for example, it can be obtained through contact between blood and sexual fluids, right? So you have to be well enough to be having intimate contact with other individuals. It can also be transmitted, so if we look at how it is transmitted, we should write that down. Okay. So transmission of HIV. Right. Blood, and we'll just put semen or vaginal fluids. Right. So if a person opens wounds, like for example on their genitalia, that could be a problem. And also bleeding due to anal sex could also be a source of contact between blood and semen. It also could be oral sex. So if you have sores in your mouth, or you have bleeding in your gums, for example, around your teeth, that could be a possible source, okay? Now, it was initially transmitted through blood transfusions, right? So even this was like early on, right? Before we knew that HIV was going to become a problem, or we even knew about HIV, people were actually getting it early on via transfusions, right? So we didn't test for that. Now we make sure and screen the blood for the presence of HIV. It can also be found in breast milk, right? So it can be transmitted to the newborn via the mother. And so that's why when people have HIV, they recommend not to breastfeed, right? And it can also be transmitted across the placenta. So if you're pregnant and you go to the doctor, they say, we would like to screen you for HIV. And you can actually say, no, I do not want to be screened. But you want to be screened because if you do have HIV, they can give you drugs that will decrease the likelihood that it'll be transmitted to your offspring. So hopefully you won't give it to the baby. And so that's across the placenta. And then we can also get it through intravenous drug use with shared needles. So that's why a lot of countries have a program where you can actually come in and get clean needles for free, because they figure the cost of having people spreading disease through the use of needles outweighs the cost of providing them with clean needles, so that you don't have a epidemic in a population that you have to treat. Okay. So those are the ways that HIV can be transmitted, right? Known transmission. Okay, so HIV, right, it's not easily transmitted. So therefore, it has to have a long latency. and a low virulence. Because normal, healthy people have to be using drugs, they have to be having babies, right? And they have to be giving blood, and they have to be having sex, right, in order for it to be transmitted. So the hope is if that we make it harder, if we even make it harder for the HIV to be transmitted, perhaps it'll even become less virulent, right? So if we practice safe sex and we clean up the blood supply and we, don't use intravenous drugs with shared needles, then perhaps we will actually see the evolution of lower virulence in the HIV itself. Okay, and then there was that idea that certain number of individuals are immune to it, and that might actually increase in the population over a given period of time, right? So that idea that there's a mutation that helps to prevent infection. Are there any questions about that video? No, okay. Okay, let's talk about the cytotoxic T cells. So the cytotoxic T cells have the ability to bind to antigen presenter cells. Okay, so they are presented with antigens, and in response, they produce two different chemicals. They produce perforins. So just think, to perforate is to create holes, to break through. So perforins are chemicals that can actually produce holes in the pathogens. And then you can also produce granzymes. And so the granzymes are produced, and these are enzymes that enter the pathogen and digest them from the inside out. Okay, so those are the cytotoxic T cells. Sometimes these are also called killer T cells, and so you'll sometimes see that term in other textbooks or online. You might see killer T cells. So these can also detect cancer cells. So remember how I mentioned that cancer is happens pretty regularly in people, but if your immune system is strong enough, you have lots of cytotoxic T cells, one can detect them and then destroy them before they develop into a cancer that would be recognizable and cause problems. So in your book, there's a diagram that shows these. So this is the target cell, okay, and it is producing the perforins. The perforins are very much like those complement proteins in that they create pores. And then the granzymes are also, through exocytosis, are also put into the cell, and then this causes the cell to rupture, okay? So that would be a mechanism of action for the cytotoxic T cells. There we go, okay. So the last thing I want to talk about are autoimmune diseases today. And so remember that this is not AIDS, which is acquired. Acquired means that it has to be from something external. So autoimmune diseases are diseases where a person's own immune system attacks normal, healthy tissue. So for some reason, the immune system doesn't recognize the tissue, and so it causes inflammation, and it also causes damage. And so a good example of this is in rheumatoid arthritis. Now, this is not your normal type of arthritis. So this would be not osteoarthritis that people get just because of aging. Rheumatoid arthritis can actually occur in children. So you could have people that, like a child that has inflamed joints, and they might have difficulty moving their joints, and they could have their immune system attacking the connective tissue in the joints and causing inflammation. And sometimes it actually causes such extreme inflammation that it causes deformities in the bones. And so you can test for rheumatoid arthritis. This would be an antibody test. So there's specific antibodies that can detect, and you can test for, that are present if a person has rheumatoid arthritis. We can also look at diseases like multiple sclerosis. And do you remember what type of cell does the multiple sclerosis, does that autoimmune system attack specifically? It's the cells that form the myelin sheath around the axons of neurons. Schwann cells, right? So this attacks Schwann cells. And so this is a neuromuscular disease. Right? So people lose the ability to walk, they are in wheelchairs, right? It can also affect their organs, internal organs too, because of the way that the nervous system innervates the different heart and digestive and all the other organs in your body, okay? There is another one that is called scleroderma. And what does derma mean? Skin. And so this causes a hardening of the skin. And so people have likened it to turning to stone, because their skin becomes inflamed and then it gets replaced with connective tissue and they lose the ability to move their joints. And so that's scleroderma. And then you can have some autoimmune diseases that have kind of systemic wide effects, and that would be like lupus. So this tends to be systemic. So that means body wide. Right? So you could have it affecting the skin, but you could also have it affecting other tissues in the body, right? Other organs in the body. Now, one thing that's really interesting about some of these diseases is that they can possibly be due to an initial infection that causes the immune system to kind of go haywire. So some have been linked to initial infections. So it could be like a bad virus, for example. And then those initial infections somehow cause the immune system to just not respond properly to the normal healthy tissue. So the immune system then starts attacking the healthy tissue.
So would it be kind of like a negative?
Rh positive and Rh negative?
No.
No.
The negative selection, where our cells like don't recognize them, or they think they're foreign.
Right. So they think they're foreign, so they're attacking them. Yep. Yep. Okay. We'll talk about that later. Okay. So I'm going to stop there for today. So we'll have our quiz on Wednesday. Yes, it's important to be able to.
I won't be at this next lab, so
Okay.
I'll turn in my lab book on Wednesday.
Okay, that'd be great. Yep, and I can have it to grade.